Today, I'm going to talk about a human disease or a human cancer we call as retinoblastoma, which is also referred as childhood eye cancer. We will learn what this disease is and what happens to the eye cells when this type of cancer develops. I must mention that this is one of the very early breakthroughs in our understanding of cancers, and now we know that the key gene involved in developing this cancer is one of the tumor suppressor genes. That is, the gene's expression is required continuously in the non-dividing cells of eyes, for example, in this case. So what happens that this gene, I will talk about what this gene I'm referring to in a while, it stops expressing itself. So why a gene stops expressing and why a gene starts expressing? I gave you in my previous discussion that it involves mutations, a change in the gene, okay, that is, it starts behaving differently. That is by way of mutation. In this particular case, what was found that it is a very good example of a chromosomal mutation. I will come to that in a while. But then let us look at what is this disease I'm referring to, retinoblastoma. So it is, as I mentioned, it's a eye cancer, which is developed. This is the normal eye. It develops here in the retina of the eye, which are light-sensitive cells here. And since this disease or this type of cancer expresses itself very early in the childhood, so therefore this name also came from the stage at which it appears, that is the childhood cancer, childhood eye cancer, and usually it is expressed in the children of less than 5 years old, One good news here is that this is curable. This is curable cancer, and 9 out of 10 children can be cured, and this can be in one eye or in both the eyes. This can occur, But again, when we talk about cure of this cancer, like any other cancer, it depends at what stage it was detected and managed. So there are routine therapies available for this cancer also, and they are very successfully employed. Okay, so that's not my discussion point. Let us look at it, why it happens. What is the main cause of this type of cancer? So this cancer involves chromosomal mutation. As you may recall that mutations at the gene level can be point mutation, deletion, point mutation, double mutations, additions, deletions of single base pair, two base pairs. But when we talk of chromosomal mutations, it involves the changing of the structure of the chromosome and loss or gain of major part of the chromosomes, in terms of deletions, for example. These kind of changes have been observed in human cells, not on a regular basis, but then wherever we have issues like diseases, whether it is the genetic disease or like cancers, and that's how this the basis of retinoblastoma was discovered that this deletion mutation takes place, in the human chromosome. I will come to that in a while. But then what is deletion mutation? As you can see, simple, a part of the chromosome is deleted. And when this part of chromosome is deleted, it is also taking the genetic material, which that is the DNA, which is here, and then it rejoins here to produce a chromosome which is smaller in size, because of the deletion of this part, this much part from the chromosome. Now, this deleted genetic material takes away many genes, or whatever genes which are lying here also go away. So it means the normal set of genes are lost when this kind of deletion takes place. Okay, so if you have understood what happens in deletion mutations in a chromosome, let us see what happens in this disease. So what was seen in the patients when they did the karyotype of the cells, tumor cells from the eye cancer tumors, so what they found that a band of chromosome was missing from 13, or the missing part was from 13 number chromosome, or 13q band from 14 chromosome was missing, So, and that was uniformly observed. That missing part was common to all the patient samples, coming from. So what was found and concluded that in a normal, healthy human being, we have the presence of both the alleles, that is, RB stands for retinoblastoma, and this RB gene codes for a protein, which I will come in a while what it does. But then in a diploid individual like human beings, we have at least two alleles of this, and a normal human being is supposed to be homozygous for this gene. Okay, so both the alleles are there, and you have the normal behavior of this gene in the eye cells, that is making this protein in the cell, which keeps the non-dividing cell in the non-dividing state. Now, if by any chance one of the allele is lost, or there is a mutation here, so it comes into the heterozygous state. That is, one allele is lost, but still one allele is there. And presence of this one allele itself is good enough to keep to make the cells behave in a normal way. Okay, but then problem comes when both the alleles are lost. Both the alleles, when they are lost, one by way of deletion, that is one possibility that how both can be missed out, that one by way of deletion, and the second allele by way of somatic mutations. So when in another allele there is a somatic mutation and one allele is deleted by chromosomal mutation, so it becomes RB minus. And when it is RB minus, it means this protein is not made, which is a tumor suppressor protein, and the cell is not able to stop itself and it starts dividing uncontrollably. Okay, so this is one way of that how both the alleles are lost. That is one by way of deletion, and another by way of somatic mutation. And another way is that when the two parents, like in a heterozygous state, mother and father both are in this state, and when they produce a child, that can inherit a scenario like this, where both the alleles are deleted or from the chromosome. Okay, so what is happening here, that this kind of genetic behavior is responsible for the loss of a very critical gene which has been identified and called as RB, retinoblastoma gene, or RBP gene, or retinoblastoma protein gene. So either it is lost by way of deletion or by way of somatic mutation in the other allele. And the result is that you lose both the alleles, and you see the expression or appearance of this cancer in the childhood. Since both the alleles are lost, this disease appears very, very early in the childhood. So if it is detected, yes, it can be cured, early. And even later, the success stories are very many for this type of cancer. Okay, then next question is what this protein or gene does to a normal cell and how it controls this whole process of retinoblastoma cancer development in human. That has also been worked out, and we have fairly clear idea about that how this protein functions. So what happens? See, this is the normal cell cycle, wherein any non-cancerous cell or normal cell is kept in a G0 phase, that is resting phase, and what keeps these cells, in this case, the retina eye cells, in G0 phase is this combination, that is the RB protein and this E2F factor. Now, E2F factor is a transcription factor which controls the expression of genes which are involved in normal cell division. So what happens? This RB protein complexes with this factor and doesn't allow it to express the genes involved in cell division, and therefore it keeps the cell in a non-dividing state. Okay, but then what happens? What happens in absence of RB? Or when this cell wants to divide? In that case, what happens? This bond is dissociated, and this protein undergoes phosphorylation, and this factor is released. So this is released, and it goes and start expressing the genes which are involved in cell division, and the cell starts dividing. So long this remains in a phosphorylated state, this RB factor, and not available to complex with this cell will keep on dividing. And that is what happens in retinoblastoma, that this protein is not available at all, and therefore this E2F factor comes into play and make these retinal cell divide without any control, Okay, but then in a normal cell, what happens? This undergoes a dephosphorylation process. If the cell doesn't want to divide, it undergoes dephosphorylation, and RB is again available here, free, and it complexes with this factor again and stops the cell division. And that is what happens in a normal scenario and in absence of RB. This RB is not available to complex with this transcription factor. And therefore, this factor then start expressing these genes which don't express in a non-dividing cell, This theory was further confirmed by in vitro experiment also, wherein the in vitro binding of this RB protein with tumor antigen like SV40T and this factor was also checked. And the same behavior they show that when it binds to this factor, E2F is free and it controls, it allows cell to divide. And when this complex is broken, the cells stop dividing, So both in vivo and vitro experiments confirmed that what RB does, and in absence of RB protein, what goes wrong, So to summarize, what I have tried to explain to you, that how a deletion mutation, if it happens and the cell become homozygous, means both the alleles are lost. The RB protein is not made. And when RB protein is not made, it is not available to complex with the transcription factor E2F, and the cell starts dividing and becomes cancerous. So thank you very much. I hope you enjoyed listening to this video as much as I enjoyed presenting it to you. Thank you very much.